This is part 3 of chapter 11A, section A, on the nervous system. Okay. We were discussing neuron processes at the end of the last lecture. Myelination in the peripheral nervous system is formed by Schwann cells that wrap around the axon of the neuron. Sorry, I lost my train of thought and I got interrupted. But the plasma membranes of those Schwann cells have less protein. So when we talk about the plasma membranes, I mean the plasma membrane of the Schwann cells that form the myelin that wraps around the axons. They have less protein, so they don't have any channels, protein channels or protein carriers. So that makes them good electrical insulators. And insulation on electrical wiring causes the electricity to flow faster. So insulation on the axons in our bodies is going to cause the electrical impulse or nerve impulse to go faster. Okay. This just shows you how the cell wraps real tightly. The cell membrane of the Schwann cell wraps really, really tightly around the axon to form the myelin sheath. This is a myelin sheath under the microscope. Okay, so here, we've seen this picture before, but now I want to show you about the gaps. Sorry. The myelin sheath gap is also called a node of Ranvier. Again, that's pronounced node of Ranvier. And so what happens is everywhere that there is, if you had a regular axon with no myelination, okay, I know I'm not drawing it exactly like the one down there, but let's just say you have an axon with no myelination. The electrical impulse has to propagate or travel all through that axon. But if you have one with myelin sheaths around it, it will skip. The impulse will skip from node to node to node. And that's the way I'm drawing it. It looks slower, but in reality, it happens much faster. So that's a faster signal because it doesn't have to travel as far. It skips over large distances of the axon. Okay. In the central nervous system, the myelin sheaths are formed by oligodendrocytes. Now, the whole cell is used in the peripheral nervous system. The entire Schwann cell forms the myelin sheath, but the oligodendrocytes just wrap around the axon. So the entire cell does not, it's just a process of the oligodendrocyte that forms the myelin sheath. You see how this is the cell and the nucleus, and it's just the processes that form the processes of the oligodendrocytes that form the myelin sheaths around the axons. And then again, we have a myelin sheath gap. That's called the same thing in the central nervous system, a node of Ranvier. In the nervous system, you will hear the terms white and gray matter. White matter consists of mostly, almost all myelinated axons. Okay, if it's gray matter, then that's going to be the dendrites, the neuron cell bodies, and unmyelinated axons. But white matter is always consists primarily of myelinated axons. Now, there are more than one type or classification of neurons. Here we go. This is a good picture. The one that you see on the left is a Purkinje cell that is found in the cerebellum. Now, if you look at the brain, like if this is your forehead, and this is the brain. The cerebellum is this part back here, is the part that helps us with balance. But a Purkinje cell has many, many, many dendrites. It's considered a multipolar cell because it has very large number of dendrites, and it has a cell body and then an axon that extends. And then you have a pyramidal cell, which is another multipolar type of cell, which has dendrites at one end and the axon at the other end. So signals are coming from here to the cell body, and then they're traveling down the axon. Don't be too confused by this pyramidal cell because it is a multipolar neuron. This axon is only showing part of the axon. It's really much, much, much longer, okay? Then you have the bipolar neurons. And what happens here is that you have a cell body that has, I don't know how to explain this to you. It basically has two projections that both have branches on each end. They both project off of the cell body, but there's one fiber here, and then it has branches, and then there's one fiber here, and it has branches. So like from here to here is the fiber that leads to the dendrites, and from here to here is the fiber that forms the axon. So it's not, see, the dendrites come off of the cell body. I mean, they are attached to the cell body in the multipolar neurons. But here, you've got a long process before you get to any dendrites, before you get to any axon terminals. You have the long processes. So that is a bipolar. And a unipolar is like the oligodendrocytes, where you have a cell body and an extension, and then a fiber that runs in both directions. The fiber that runs toward these branches is going to act, it is an axon, but it's still going to receive impulses and then transmit them to the other end, which is also an axon. So that's a unipolar neuron. So we've got multipolar, bipolar, unipolar. I didn't know if they were going to show you. And this is good where you can see them, the whole entire thing. I should have shown you this way. Multipolar. Lots and lots of dendrites attached to the cell body. Long axon. This axon in particular is myelinated. And then you have branches that form the axon terminals. The bipolar neurons have a cell body that have an extension going in both directions away from the cell. The one on the left is called the dendrite because it has a lot of branches and the signal travels toward the cell body. The electrical signal travels toward the cell body. And the axon does have branches on the other end, and the signal travels away from the cell body. So that's how you know the difference. And then you have the unipolar, which there is nothing directly attached to the cell. The cell is attached to a long fiber. The whole thing is called an axon. That whole axon, in this case you see it is myelinated, has branches at one end that receive a signal and branches at the other end that transmit a signal. Okay, so sensory neurons transmit impulses from sensory receptors toward the CNS. They are almost all unipolar. Their cell bodies are located in ganglia in the peripheral nervous system. Motor neurons carry impulses from the central nervous system to the effectors or the targets. They are multipolar, all of them, and most of the cell bodies are located in the central nervous system, within the central nervous system. Interneurons, which are also called association neurons, lie in between motor and sensory neurons, and they help to integrate the information from the sensory and use it to determine what signals to send to the motor neurons. Most interneurons are entirely within the central nervous system, brain and spinal cord. And this is just showing you some examples. A motor neuron that's attached to a skeletal muscle is multipolar. A bipolar neuron, for example, found in the retina of the eye, the rods and the cones um, that sense vision and then transmit those signals to the optic nerve, which carries the information to the, visual center in the occipital region of the brain. And then there's a sensory neuron that senses touch and senses either touch or pain or hot or cold, some kind of sense in the skin, and transmits that to the spinal cord, to the dorsal region of the spinal cord. That'll be important later. And that is an example of a unipolar neuron that does that. Okay, now we're going to move on to part B, the membrane potentials. Now all cells, even neurons and all other cells, have what's called a resting membrane potential. This is the outside of the cell. This is the plasma membrane. The plasma membrane has a slightly more negative charge voltage. I'm just putting this to represent negative charge than the outside, which has a slightly positive charge. So if you took a voltmeter, a very, very, very like microscopic voltmeter, and you measured the voltage from here to here, that would be the resting membrane potential of the plasma membrane. In neurons, that resting membrane potential is negative 70 millivolts. And what maintains that resting membrane potential is you may have heard this before, especially in Bio 111, the sodium-potassium pump, which we will go over that a little bit more too. Sodium, that's an A, sodium-potassium pump. Okay, so we know neurons are highly excitable, and it begins with having a negative 70 millivolt resting membrane potential or voltage, okay? Because they have to change that voltage to a positive voltage in order to generate an action potential. So if you don't start with negative 70, you can't generate an action potential at all. So that resting membrane potential is necessary for you to have a signal transmitted, okay, from one place to another. Basic principles of electricity. Opposite charges are attracted to each other. Negative charges are attracted to positive. Energy is required to keep opposite charges separated across a membrane, and that energy comes from the sodium, potassium pump. And the energy is liberated or freed or released when the charges move toward one another, and when opposite charges are separated, the system has stored energy. All right, so voltage is the measure of potential energy. You will hear me use that interchangeably. I might say resting potential or resting voltage. The voltage is actually the units of measurement. We are using millivolts in the human body because we don't have real high voltages, but we do have potential energy. We do have voltage. We do carry electricity. Okay, so the greater the charge difference between the points across the plasma membrane. So here's again your cell between the 2 points of the voltmeter, here and here. The greater this distance is from one to the other, the higher the voltage, the greater the charge difference. So if this is negative 70 millivolts and this is 30 millivolts, that's a big charge difference. But if this was negative 10 and this was negative 11, that's a small charge difference. Okay, so the greater the difference in charge across the membrane, the higher the voltage is. The current is a flow of electrical charge, and resistance is what happens when you have an insulator or a conductor. It's a hindrance to the flow of charge and we will talk about this more in the next section.